Dystonia is a complex movement disorder that affects people of all ages, causing involuntary muscle contractions, abnormal postures, and sometimes tremors or jerky movements. Its clinical presentation is highly variable, making diagnosis challenging, especially outside specialized centers. Dystonia can appear on its own or as part of broader neurological syndromes, adding to its complexity. Historically, dystonia has been recognized since antiquity, but only in the 20th century did formal clinical definitions emerge. Today, experts define dystonia as abnormal movements or postures, often twisting but sometimes tremulous or jerky, that may occur independently or together. Understanding dystonia requires a look at its diverse causes and the intricate networks within the brain that control movement. Diagnosing dystonia is not straightforward. Its symptoms can vary widely in age of onset, affected body regions, and progression over time. Some patients may show clear abnormalities on brain MRI, but most do not, which complicates the diagnostic process. Instead, current models suggest dystonia is a disorder of motor network connectivity, meaning the problem lies in how different parts of the brain communicate to control movement. This complexity means that even experienced clinicians may struggle to identify dystonia, especially when it appears as part of a broader syndrome. Recent advances in genetic research have revolutionized our understanding of dystonia. High-throughput sequencing technologies have enabled scientists to identify over 200 distinct genetic causes of dystonia, many of which are crucial for brain development. This genetic diversity explains why dystonia can look so different from one patient to another. Some genetic variants are extremely rare, while others are linked to broader syndrome. The discovery of these genetic underpinnings is helping researchers classify patients into biologically defined subgroups, paving the way for more personalized treatments in the future. Today, most treatments for dystonia focus on managing symptoms rather than addressing the root cause. Options include botulinum toxin injections, various medications, surgical interventions like deep brain stimulation, and supportive physical therapies. While these approaches can provide relief, they do not modify the underlying disease process. There is a significant unmet need for disease-modifying therapies. Excitingly, new gene-based and small molecule therapies are emerging, but their success will depend on identifying precise molecular targets and tailoring interventions to each patient's unique biology. Recent research has uncovered several key molecular pathways involved in dystonia. These include altered gene expression, impaired protein degradation, nuclear envelope dysfunction, and mitochondrial abnormalities. Each of these pathways contributes to the development and progression of dystonia in different ways. By understanding how these molecular mechanisms interact, scientists hope to identify new therapeutic targets and develop more effective treatment. This molecular approach marks a shift from treating symptoms to addressing the root causes of dystonia. Tightly controlled pattern of gene expression is vital for healthy brain development. In dystonia, disruptions in this process, known as aberrant transcriptional regulation, can lead to abnormal movement patterns. Genes like THAP1, YY1, KMT2B, and MORC2 play crucial roles in regulating gene expression. Mutations in these genes can cause isolated or complex forms of dystonia, often with additional neurological symptoms. Recent discoveries show that these genes interact closely, and their dysfunction can disrupt entire networks of gene regulation, highlighting the importance of transcriptional control in dystonia. Epigenetic changes, chemical modifications that affect gene activity without altering DNA sequence, are increasingly recognized in dystonia. For example, unique DNA methylation patterns or episignatures have been found in patients with mutations in KMT2B, YY1, and MORC2. These episignatures can help reclassify ambiguous genetic variants and even predict disease onset. The field of epigenetics is opening new doors for understanding why similar genetic changes can lead to different symptoms, and it may soon provide new biomarkers for diagnosis and prognosis. Cells rely on precise protein quality control systems to maintain health. The proteasome, a complex that degrades unwanted proteins, is central to this process. In dystonia, recent studies have shown that mutations in genes like THAP1 and FBXO31 can impair proteasome function, leading to the accumulation of damaged proteins. This breakdown in protein turnover is now recognized as a key mechanism in some forms of dystonia. Understanding how these pathways fail could lead to new treatments that restore proper protein degradation and cellular balance. Another important player in protein regulation is AOPEP, an aminopeptidase involved in processing polypeptides and maintaining proteome homeostasis. Mutations in AOPEP have been linked to isolated dystonia, and the protein is especially abundant in glial cells like oligodendrocytes, which are crucial for myelination. This connection highlights how disruptions in protein processing and myelination can converge to produce dystonic symptoms. Ongoing research is exploring how targeting aminopeptidase activity could offer new therapeutic options. Nuclear envelope, NE, separates genetic material from the rest of the cell and is essential for proper cellular function. In dystonia, dysfunction of the NE has been implicated, particularly through mutations in genes like TOR1A, NUP54, and NUP62. These genes are involved in maintaining NE integrity and regulating the transport of molecules between the nucleus and cytoplasm. Disruption of these processes can lead to abnormal protein trafficking, stress responses, and ultimately, movement abnormalities. This area of research is uncovering new mechanisms that may be targeted in future therapies. Mitochondria are the powerhouses of the cell, providing the energy needed for all cellular activities. Some forms of dystonia are now linked to mitochondrial dysfunction, especially defects in complex V of the oxidative phosphorylation system. Genes like MT-ATP6, ATP5MC3, ATP5F1A, and ATP5F1B have been implicated. When these genes are mutated, energy production falters, particularly in neurons with high energy demand. This can lead to a range of neurological symptoms, including dystonia. Understanding mitochondrial involvement is opening new avenues for treatment, including the use of compounds that boost mitochondrial function. With a deeper understanding of dystonia's molecular mechanisms, researchers are exploring innovative therapies. Potential strategies include targeting DNA methylation with specific inhibitors, enhancing proteasome activity, modulating nuclear cytoplasmic transport, and boosting mitochondrial function. Some of these approaches are inspired by treatments used in cancer or neurodegenerative diseases, such as DNMT inhibitors, proteasome activators, and mitochondrial boosters like CoQ10. While many of these therapies are still experimental, they represent hope for more effective, disease-modifying treatments in the future. One of the most promising developments in dystonia research is the move toward patient stratification based on molecular profiles. By grouping patients according to their specific genetic and molecular characteristics, clinicians can tailor treatments more effectively. This approach also allows for better prediction of disease progression and response to therapy. Large-scale sequencing, deep phenotyping, and long-term follow-up are essential for refining these strategies and moving toward truly personalized medicine in dystonia. Despite significant progress, many challenges remain. The vast genetic and phenotypic heterogeneity of dystonia makes it difficult to predict disease course or response to treatment. Incomplete penetrance, where not all individuals with a mutation develop symptoms, adds another layer of complexity. Researchers are now investigating how environmental factors interact with genetic risk to trigger or protect against dystonia. Continued collaboration across disciplines and the integration of new technologies will be key to overcoming these hurdles. Future of dystonia care lies in personalized medicine. As researchers unravel the molecular networks underlying dystonia, the disorder is being transformed from a clinically defined syndrome to a spectrum of biologically distinct diseases. This shift promises more accurate diagnoses, better prognostic tools, and targeted therapies tailored to each patient's unique biology. While many approaches are still in development, the growing understanding of dystonia's molecular basis offers real hope for improved outcomes and quality of life for patients worldwide.